Hello everyone, my name is Sekhar. I'm a PhD candidate at Dr. Man's lab. So today I will be talking about a first meta-analysis done in the field of animal behavior research. So this project is all about ungeneralizable generalizations, the influence of taxonomic bias on meta-analytical inferences in behavior, which I recently presented at the annual Animal Behavior Society meeting in 2025, where it got recognized with the Founders Memorial Award for the best poster. And this project is in collaboration with Professor Shinichi Nakagawa and Professor Ned Doctorman. So traditionally, behavioral research has focused very strongly on vertebrates, while invertebrates remains underrepresented. For example, there was a paper published in 2017 by Rosenthal and colleagues, where they showed a clear taxonomic bias in animal behavior publications, with a vast majority of the studies focusing on mammals and birds, while insects, crustaceans, and other invertebrates were relatively neglected. Often, in order to draw the broad conclusions and identify general patterns in the underlying research, the researchers mainly rely on meta-analysis that synthesize the result across these studies. Yet, if the invertebrates are consistently underrepresented in these data sets, the resulting estimates may reflect vertebrate patterns disproportionately, limiting the generality of the conclusions. This imbalance is important, and often we don't talk about this imbalance because this imbalance can cause a major problem in the effect sizes that goes into the meta-analysis. And these meta-analyses draw the conclusion based on whatever effect sizes actually goes into their data. So if the data itself is highly biased towards vertebrates as compared to invertebrates, then the general conclusion which we are drawing out of these meta-analysis also need to be interpreted with a lot of caution. In other words, our ability to make general conclusions about the animal behavior could be compromised if our samples are biased. So in this study, we asked two questions. First, is the taxonomic bias reported in the behavioral research also reflected in the behavioral meta-analysis? And the second question, does the taxonomic bias in the sampling affect the effect size estimates produced in those meta-analyses? Based on the prior work, we predicted two things. One, that the data used in the behavioral meta-analysis would mirror the broader taxonomic bias, meaning vertebrates would dominate and invertebrates will be less represented. And the second one, the effect size would vary across the animal groups, such that the meta-analytical estimates and the inferences drawn from them would themselves be taxonomically biased. So let's talk about the methods in detail. To test these questions and our prediction, we search for meta-analysis published in three major behavioral journals: Animal Behavior, Behavioral Ecology, and Behavioral Ecology and Sociobiology from 2000 to 2024. So the initial search gives us almost like 118 meta-analysis. And based on certain inclusion criteria, we narrow it down to 15 papers. So the inclusion criteria include there must be a meta-analysis and it should be a non-human subject, must be for behaviors and not taxonomically restricted. Once we have all these data, next we reran the published meta-analysis to estimate the overall effects based on the actual representation. To evaluate the influence of taxonomic bias, we generated marginalized means for each analysis, which essentially recalculate the result as if 95% of the data had come from invertebrates. We then compared these observed estimate to the adjusted ones, quantifying the differences as absolute log ratios. Finally, we used a random effects meta-analysis to estimate the overall magnitude of bias. Now let's move to the results portion. Before I talk about the results, I want everyone to have a closer look at figure 1 and figure 2. So figure 1 basically talks about the Rosenthal work which they did in 2017. As you can see, around 90% of the data is of vertebrates. We would expect the other way around. We should expect that most of the work should come from an invertebrate. But actually, this is what happened. And it's quite surprising. And the figure 2 represent almost the same misrepresentation of vertebrates versus invertebrates, even in meta-analysis. So let's talk about the results in general. Just as in the primary behavior literature, we found that the data used in meta-analysis is strongly taxonomically biased. About 65% of all effect size estimate came from vertebrates, leaving invertebrates highly underrepresented. You can understand that from figure 2 of this poster. This bias had a meaningful consequences. Across studies, the overall mean effect size was estimated at 0.30 with a standard error of 0.02, which was highly significant. But when we adjusted for taxonomic bias, the estimate shifted substantially. In fact, on an average, about 35% away from the observed values. So if you look at the other panel, these are the papers which we used for our meta-analysis. If we would have sampled based on the original taxonomic representation, the things would have been much, much different. In fact, the sign of the effect changed for 3 of the 43 analyses, and the statistical significance changed for 10 of the 43, or roughly 23%. This means nearly 1 in 4 published conclusions could have been different if the taxonomic bias had been accounted for. We also observed moderate heterogeneity both between and within studies. To demonstrate how bad this can impact the overall conclusion, I wanted to talk one work which published from our own lab, which is the Roytes paper in 2018 on the pace of life syndrome. So this paper was published in 2018, and what we have found was there was an effect size of 0.06. But when we correct that for taxonomic bias, the estimate is increased to 0.22, which suggests a stronger support for the pace of life syndrome. This is not just a minor change. It alters the major conclusion of the paper. So what does this mean for the behavior research? Our findings show that taxonomic bias strongly influences the generalizability of meta-analysis. In other words, the conclusions we draw from the published synthesis are not as broadly applicable as we might think. This might have several implications. First, the taxonomic bias in behavioral studies need to be directly addressed if our goal is to build a general theory of behavior. That means prioritizing the inclusion of a wider range of taxa in primary studies would be essential and crucial, especially for invertebrates. Second, meta-analysis, or those who are doing meta-analysis, should make more use of subgroup analysis and marginal estimation, which, although they may yield different results, can provide more robust and representative inferences. Third, the journals and reviewers should encourage the authors to explicitly report the taxonomic breakdown of their data to discuss how representation might influence their conclusions. So, transparency is the most important key thing in these kind of situations. So, based on our research, to conclude, what we have found is the behavioral meta-analysis are strongly biased towards vertebrates, and this bias substantially influenced the effect size estimates and statistical significance and the interpretation of the results. Addressing this issue will require efforts both at the level of primary data collection and at the level of synthesis. With that, I would like to conclude, and I wanted to thank the Environmental and Conservation Science program at North Dakota State University and the Department of Biological Science for funding. Professor Ned Dr. Mann is supported by NSF grant. And with that, thank you all for listening, and I look forward to your questions.